Okay, so back in the protein editor again, what other things do we have in the frame? Well, we have synonyms, and that's just where you can put in names. It's important that you populate this if you know synonyms because these become searchable when you search your database. And so it's a way for you to find a protein by a different name when it comes up in the literature, a name you're used to but your neighbor in the lab is not, that kind of thing. General citations. Now, we like to use a format for the summary of what we like to call assertion citation. And that means that we intersperse the citations within the text, within the free text here, much in the same way that you would in the discussion section of a paper. And we also like to have sort of one-liner that explains the function up top without going into too much detail or having a citation. Now, I and many of my fellow curators normally do these kind of descriptions. We type them up first offline, then paste the whole thing in here. But what you might imagine having is something like this, where we say that aspartate kinase carries out key steps in lysine and homoserine biosynthesis. And that's the sort of general overview sentence. And then you might have something like, and one of the things I like to do, I'll put it here, is put the protein name there in the back so that I don't have to go along saying aspartate kinase every time I want to reference it. I can just say lyc. And then here I can say lyc catalyzes, blah, 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 where I describe a specific function. Then I might want a citation right after that, in the sentence, the way that you would do it in the discussion section of the paper. So I have the cursor where I want it, and I go over here and I click this thing that says CITS, so CITES, it's our citation system. And I click that, and it makes a ready-made citation slot, and I can type in, again, a PubMed ID or free text if there's no associated PubMed ID. And then I can have a second sentence giving more detail, and then a third, and then maybe another citation after that, right? And you notice this will just keep scrolling sideways, so sometimes it's nice to just introduce a line break yourself. Line breaks like that, if you hit return here and it continues over there, those are actually invisible to the software, and you'll see what I mean in a moment. So let's put in this citation again. I know naturally a real entry will probably have multiple paragraphs if there's a lot of detail without the same thing cited over and over and over again. But let's hit OK. And so now we have lyc catalyzes blah, blah, blah. We have our first sentence here. And you notice even though there was a line break in the middle here, it disappears when this is put up here. Paragraphs in our summary boxes are separated by full lines. Full lines in between gives you a new paragraph. Other individual line breaks are meaningless. So let's save our expert curation here. And of course, this is just a really quick overview of how to put together a summary like this, and more on the technical side and not on the formatting side. You're free to, of course, put in whatever you want. You could have a laundry list of sentences here with a citation after each one. You can do whatever. We find that it's easier to read if it flows and it has structure and it reads like a nice encyclopedia entry. Now going back in one more time, there is one other thing of major note, well, two other things that you can do here. You can add frames and hyperlinks. Now a hyperlink is exactly that. It's going to go out to an outside URL and it will open up in the appropriate web browser, and you can add a frame, and that might be if you said something like lyc interacts with, and then when you click on frame, in this case it's going to give me a big view of things I've looked at recently. So let's pick protein rec A, and it puts the frame ID in, which is great because it means I didn't have to click over to the frame, look at the frame ID, remember this, and write it all down. However, if I had wanted to go over the protein and look at the frame ID and write it all down myself, I could again click on frame and just click away, and now it puts in an empty frame setting for me. And I'll just get rid of that because I don't feel like doing that right now. And the last thing here is that you can use the spell check to do exactly that, check your spelling. But let's close this for one more moment. And this shows you what happens when you put in a frame like that. You get a thing that says sentence, 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 the frame reference where it puts in the name of the frame. And that's actually now a clickable link to that frame. And again, as I'm a habitual saver, we'll save one more time. Okay, so let's go back to the editor one more time. Just click here, Edit, Protein Editor, and let's take a look at what's left in a protein frame that we might want to add or modify. Okay, so we can indicate locations for the protein, and this takes you to our whole cell component ontology, and you can pick all your familiar prokaryotic and eukaryotic locations. Let's just close that. You can link out to other databases. We have some pre-made ones you might want to use, like linking out to, say, PubChem or, go down here, Uniprot, etc. You can, now this is a very important one, you can do a credit system where individual authors are credited, and you can click here to update a last curated date. And this can be really handy when you want to keep track of the last time something was curated and you want to do it in a fairly definite manner. Now in the past, before we added this feature, we'd do things like look at what the most recent paper was that was included in the curation. But that's not always indicative. The most recent paper might well be from 1975. And in that case, you want to know that you don't have to keep going back and checking the entry again and again. When you click the last curated date, you'll get a marker, which we'll see once I leave here, but has today's date, which happens to be the 15th of November 2007, that says the last time someone gave this a thorough looking over. And so the next time someone looks at it, they don't really need to go back farther in the literature, for example. Down at the bottom, we have the ability to edit the enzymatic reaction frame. This is actually a different frame, a frame that links the protein to its reaction. And if you want a little more detail about what's going on there, you can look at our introductory webinar a little bit, and you can look at our documentation. And in this frame, you can add an evidence code specifically assigning why you think this enzyme does this reaction. So again, we could say, oh, we saw it in the purified protein, and you can add a citation. And here you can mark the reaction direction. The default assumption is that the reaction direction is unknown, but you can indicate that it's reversible or unidirectional. And in more up-to-date versions of pathway tools like the one that we're about to release, you can actually add a citation in here as well. Then down below you can put summaries of things specific to the enzyme activity. We often put things like KIs for certain inhibitors in there. So it's not general protein stuff, but stuff specific to the activity with all the usual summary tools. And then here there's a structured way to put in activators, inhibitors, and cofactors with all these different possible types. And so what you might do is you put in, okay, there's an activator. Let's say that perhaps our activator is divalent magnesium ion, though that's more likely to be a cofactor. Is it physiologically relevant? And we'll say yes. And what this means is that it will actually show up, the regulation will show up in pathway level displays. You know, for example, feedback inhibition is done that way, where it's inhibition, the compound, and it's physiologically relevant, and you can put in a citation. Then down at the bottom, you can put in things like optimum temperature, optimum pH, and you can put these in as ranges. So you can do something like pH 5 to 7, and it will convert it into this 5:7, but that just means pH 5 to 7, or we could have had it be 5 to 7.5, or whatever, or just a single number. And then KMs for all of the compounds involved. And these KM fields will just automatically appear for the relevant compounds based on the reaction equation up here. And so now you know how to fully populate a protein frame. And if we click down, you'll see I have an activator. It says it's a physiological regulator because I said it was, even though I was weird and chose magnesium, which is more of a cofactor. And I'll hit save again.